Welcome to the Functional Medicine Doc Talk podcast. I'm your host, Dr. Kurt Woeller, talking with experts in functional and integrative health and medicine, discussing critical information for improving your health and wellness to help you live a long, full life. Let's get to it. Hi, everybody. Welcome to the Functional Medicine Doc Talk podcast. I'm your host, Dr. Kurt Woeller. And I'm joined today by Dr. Chandler Marrs. We're going to have an interesting discussion about thiamine, or vitamin B1. And Dr. Marrs is actually an author of a book. I'll tell a little bit of story about that book and the influence it had on me with regards to some of my study into biochemistry, etc. So, a lot to talk about. So, Chandler, welcome to the program.
Oh, thank you. I'm glad to be here.
So let's go ahead and get into it. Real briefly, prior to us, getting on here and doing a recording, you mentioned that your PhD is in experimental psychology, which seems a bit of a maybe distant from this discussion about thiamine. But I want to know more about how you got interested in thiamine, because the information you provide with regards to thiamine, the biochemistry, the applicability of it, is very extensive. So give me a little background about how you got interested in this work.
So I am someone who tends to think outside the box in everything I do and tends to dig deeper in just about everything I do. So with my interest in neuroendocrinology, which is what I began, hormones in the brain, I was always very interested in finding the pathways, the various hormones that were linked to various cognitive and behavioral effects. And I dug and dug and dug and illustrated some patterns in my dissertation. But it was outside of the box, and it was not stuff that was readily accepted. And frankly, the 2009 crash hit, and I'm in Vegas. And so I became unemployed and had to begin to start over. Had children, couldn't move. Began writing on my own blog, Hormones Matter, because that was my original interest, was hormones. And as an advocate for women's health, went down a number of pathways towards medication adverse events, obviously hormone issues, and so on and so forth, and landed in the Gardasil vaccine controversy. And in working with the Gardasil vaccine, with patients who had been injured by the Gardasil vaccine, the research led me to the mitochondria. And as a Ph.D. who had done some biochemistry, but had done mostly, endocrinology and the like, when we talked about mitochondria, it was just as little energy factories. They just kept working unless you had alcoholism or starvation or some rare genetic disorders. Mitochondria did the things they were going to do. Well, it turns out that that's not the case. It turns out that they actually need nutrients, and they can be easily deprived of those nutrients from, certainly diet, but from medications that block nutrient pathways, but also medications can damage the mitochondria in a variety of ways. And so I began exploring those things. And through a series of connections, a parent, frankly, of a young girl who was damaged by the vaccine, connected me with Dr. Lonsdale, because the mom had decided that her daughter suffered from thiamine deficiency. And as anyone who knows about thiamine deficiency, Dr. Lonsdale is kind of the leading expert in thiamine deficiency. And she connected him to me, and we began working together. And this was, about 10 years ago. And gradually, I became more versed in thiamine, and of course, started digging into the various pathways and trying to determine why so many people were deficient in thiamine. Because I was taught, unless you were an alcoholic or unless you were starving, you were not likely to become deficient. And yet I was seeing thiamine deficient everywhere. And so that's how I started.
It's interesting about the mitochondria. I'm actually showing your book that came up. And so, I was thinking about prior to this interview, so I've been in practice for 25 plus years. And like any doctor, I read, I research. Usually it is at the sort of the impulse, if you will, of a patient that comes to see you who might have a particular condition. And so you read about that condition. And I've always read, but I've read a lot of different things over the years, from delving into history to music to, of course, medicine and whatnot. And I got to a point, probably, I want to say 5, 6 years ago, maybe around there, where I thought, I need to go back to understanding more of the fundamentals of things. And so I don't know when your book was published, the Thiamine Deficiency Disease, but when I was thinking back and reflecting on this, your book with Dr. Lonsdale was something that really was a pivot point for me. In that, when I read your book and I started looking at thiamine with regards to the different enzymes that it influences and how that is involved in mitochondrial activity, it was a spark for me to think, okay, I need to now go back and study biochemistry. You get biochemistry in medical school, but you sort of pass through it because you're just inundated with stuff. And so, it was around this time, but I wanted you to know that your book was a pivotal point in my journey. So from that point, I've read textbooks on biochemistry, on immunology, on endocrinology, and inborn errors of metabolism. I'm constantly evolving. And as you know, I do a lot of work with organic acid testing and teach seminars on that. And you'll realize even within organic acid metabolism, a lot of it is related to thiamine. So it's interesting in that your journey took you sort of kind of to a similar place that brought me in, and going back and looking at the biochemistry. What I find interesting, just purely from a psychological standpoint, if we think of the brain and nervous system, uses uses up about what, 20%, maybe 25% of all the ATP that's produced.
That's what they say.
On just a moment-to-moment basis. So the brain and nervous system is a huge drain, if you will, on energy reserves. And so it needs a lot of mitochondrial activity in order for it to get generated. So very interesting. And then with regards to the Gardasil and the dysautonomia that tends to trigger, I'm curious, in that particular case, do you remember how quickly the onset of that child's issues were?
Goodness, it was years ago. It was pretty quickly. And I don't remember if it was after like the first or second or there's 3 doses. And a lot of the young girls and young women would be okay. And I put that in air quotes because you're never quite okay after the first dose. And it was often the second or third dose where everything just hit. And it's pretty much a toxic overload, frankly.
Right.
And while I'd like to think that Gardasil is unique in its ability to damage a variety of systems, over the years I've learned that it's not, that all pharmaceuticals, vaccine or drug related, will hit the mitochondria by one mechanism or another. And once you start seeing those patterns, you know that not only is it blocking certain pathways, but it's damaging energy production, ATP. And when you damage energy production, then your capacity to withstand any stressor becomes diminished. And so that's kind of the cascade of events that begins. And whether it is a direct blockage of thiamine, which a lot of pharmaceuticals will block thiamine transporters, it's very common, or it's just a blockage of another pathway, you're always going to need more thiamine because you're going to need more energy to withstand and navigate your body to navigate that stress, so that it doesn't become this endless cycle of poor capacity, more toxins, poor capacity, more toxins, and so on. And so thiamine is integral to all of that. And you think, well, certainly it's not like a magic pill, but in some regards it is, because it is the rate limiting step to those energy pathways.
Before we talk about the energy pathways, and specifically I want to get into some of the enzyme reactions, but in traditional nutrition, right, we've seen the discussion in the literature of like, well, in a modern world, with refined foods, people aren't having thiamine deficiencies, so it's not a big deal, don't worry about it. And so the levels or the amounts of thiamine, and essentially to get an RDA standard, or what would be considered to be a sufficient amount, is really not a lot.
No, it's a single milligram.
Yeah.
I mean,
So what, and maybe the question's obvious, right? Comparing our world today versus 100 years ago, 150 years ago, just the food, certainly the chemicals and whatnot, certainly all of that can play a role, and I think certainly does. But is there something unique about thiamine, its chemical nature, its structure, that allows it for it to get interfered with, to get broken down, to something to inhibit its function? Is there something unique about it?
Well, there's
versus maybe
there's
other
a number of
other
things,
items?
but there's also structural. Let's back up.
Sure.
Is it the structure of thiamine that is unique relative to the structure of all the other vitamins? Yes and no. What's unique about thiamine is not that it can be broken down by so many different foods and medications and so on and so forth, because all the vitamins can be. I mean, to some extent or another. What's unique about it is its position, its gateway position to the entry into oxidative phosphorylation, which is the TCA cycle in the mitochondria. So if you don't have enough thiamine, it's in multiple pathways. But this key one, the pyruvate dehydrogenase enzyme, this key enzyme is the gatekeeper. And if you don't have enough thiamine, there is nothing getting in there. And so that cycle that is going to get us, from A to B through the electron transport chain out to ATP is not going to work. And so that's the key issue here is the pyruvate dehydrogenase enzyme. Now, there are also other enzymes there's enzymes in each of the pathways, that involve thiamine. So the PDH is the glucose one. And from the PDH enzyme and from the glucose, for each molecule of glucose, we can get 30 to 38 units of ATP. And if we don't get that glucose funneled through the mitochondria and out the electron transport chain into ATP, then it all backs up at the gate, so to speak. And then it goes off into secondary and tertiary pathways that then produce a little bit of energy, but they produce more toxicants than you can deal with. And so you've created a net deficit by just not getting into, because of all the side pathways it has to go to. So there's one component of it. Now, it's also rate limiting in fatty acid metabolism up in alpha oxidation. Alpha oxidation is the first process before you get to beta oxidation, before it then pops into the mitochondria. And so if alpha oxidation doesn't work, again, because it's so high up in this process, the rest of it's going to get clogged up down below. Fatty acid molecules, you can produce over 100 units of ATP per fatty acid molecule. But if it doesn't get into the mitochondria, again, you get dyslipidemias and all sorts of other things, because it has to be metabolized in these size pathways. Same thing happens with proteins and amino acids, because it's involved in what's called the branched chain keto acid dehydrogenase enzyme. Basically breaks down the branched chain amino acids. If you don't get through that pathway, you get increased uric acid, but you also get a problem with some fatty acids on the other side. And so again, all of these limit mitochondrial capacity to take the food that we consume and make it into energy. So if your thiamine is insufficient, then all of this stuff is not only going to reduce the capacity to produce ATP, but it's going to demand more ATP to process the crap that it's spitting out to compensate. So yes, thiamine is unique, but it's not unique just because of its structure. It's unique because of its position, its rate limiting position that controls everything else.
I think that's really important. I appreciate you mentioning that about, not necessarily its structure. Yeah, that's one thing, but the position and where it works. Mention real quickly the aspect of thiamine regarding the pentose phosphate pathway.
Yeah, so I didn't. Yeah, that's the other pathway. Pentose phosphate pathway is the one that produces, ribose for RNA and DNA. It produces some substrates for myelin and steroids, and for glutathione and thioredoxin. So it's a connector pathway that takes glucose and does some stuff for cells to grow and do their thing. But then it also will feed back into the PDH and process the rest of the way. Now, some interesting things about the transketolase pathway. Transketolase is the enzyme that is positioned twice in the pentose phosphate pathway. It is thiamine dependent. It is the rate limiting thing for that whole pathway. Some interesting things about the transketolase pathway is that it can be affected by anti-thiamine factors quite significantly. And this has only been recently looked at in kidney disease, chronic kidney disease, where things are going pretty bad at this point. The body produces something called oxythiamine. And oxythiamine is a competitive antagonist to thiamine, but at the transketolase enzyme. And so it sits in there, and thiamine can't get in there. Now, for the longest time, we've used oxythiamine in research protocols to induce thiamine deficiency in animals. Turns out the body produces its own oxythiamine under certain circumstances, so that, particularly in kidney patients, their thiamine concentrations look completely sufficient, even high. But it turned out they were producing high concentrations of oxythiamine, so none of it was doing what it was supposed to do. So it's an anti-thiamine factor. So in those situations, the person produces, thiamine blocking agents endogenously. Now, what are those constraints that allow you to produce that? Well, high fructose diets and poor kidney function. And what is most in every food that we eat? High fructose corn syrup.
Right.
So,
wow.
so thiamine, again, it shuts things down. And there's other connections. If you want to talk about other food-related things that are going to break down thiamine, you have the things that are going to break down the metabolism of thiamine or the activation of thiamine, like coffees, teas, alcohol, certainly. But you also have things like soybean oils, the vegetable oils that we consume that's in everything. All of those activate another enzyme, an enzyme called pyruvate dehydrogenase kinase, which is an enzyme that shuts down the pyruvate dehydrogenase enzyme and then shuts down the whole OXPHOS cycle. And so the food we eat today is just killing us by multiple mechanisms.
So that's interesting about the pyruvate dehydrogenase kinase.
Yes.
I recently did a Substack article on the effect of melatonin. So intracellular melatonin, which about 95% of melatonin is actually produced in our mitochondria, not our pineal gland. And one of the things that melatonin does is it puts the brakes on that pyruvate dehydrogenase kinase.
Oh, I didn't know that.
That releases the pressure off of pyruvate dehydrogenase complex. So, and in fact, that mechanism, hypoxia-inducible factor alpha and a few other things that get generated in aerobic glycolysis, which is the Warburg effect, because they've looked at this sort of thing in relationship to cancer. The major impact is those things increase pyruvate dehydrogenase kinase, which inhibits pyruvate dehydrogenase complex reactivity. So basically what you're doing is you're shutting off the ability to convert pyruvate to acetyl coenzyme A.
Yeah.
In essence, it's almost like, it's not the same thing, but it's almost like a thiamine problem in that you can't make that conversion. So you're just stuck in lactic acidosis. So, which is why, there's so many things that induce lactic acidosis, one of those being thiamine, but you can also have other factors. I actually read somewhere, maybe that a thiamine deficiency, or maybe it's not sort of the right form of thiamine, whatever, can actually be an inducer of hypoxia-inducible factor alpha, which puts the brakes on the pyruvate dehydrogenase kinase.
Okay, say that again. Wait.
So I can't remember where I read this, and I was maybe I thought maybe you might know, or maybe I can see if I can find the information for you, but I read that a thiamine deficiency can actually be
Oh,
an
okay.
inducer of hypoxia
It
inducible
is. It
factor
is.
alpha.
It
Yeah.
is. There was a great article published, I think, in 2013 on that. The lab is Zaster. The primary author, I can't remember off the top of my head, but I've cited it on a number of occasions. Yes, thiamine deficiency can induce HIF proteins.
Okay.
So, and the PDH kinase also induces the HIF proteins. So if you give thiamine, even if they're technically not thiamine deficiency, you can end that cycle and reduce the molecular hypoxia.
Right. Very
And shut
interesting.
off the, yeah.
So one of the things about thiamine that seems really intriguing is when I read biochemistry or read about molecular biology or cellular mechanisms, I'm always thinking to myself, how did this system develop and why did it develop? From just an evolutionary standpoint, being able to compensate for different things or overcome some type of stressor. And so we know that, and I learned this from you from the paper, which I recently read, and I want to show that paper here in a second, that thiamine can actually get into the body in large enough concentrations by passive diffusion. But if the levels are too low, then it becomes dependent on transporting mechanisms. And there's a lot of different transporting mechanisms. Maybe you can just touch on that piece a bit of the different transporting mechanisms, because it's getting across the cell membrane, it's getting into the mitochondria, all of these different factors, which is quite fascinating.
Well, in all the different transporters, let's back up. So when it's low, it requires transporters. Transporters will tend to upregulate a little bit to kind of suck more in. But there's medications, there's a lot of medications that block transporters. So you have the dual purpose or dual factors of someone having perhaps insufficient thiamine to begin with, put on a medication that blocks a transporter, and then that sends them into acute deficiency. And that happens quite frequently with flagell being one of them. One of the key side effects is thiamine deficiency, although it's never ever recognized. It's been published for years, but it blocks the transporter. Several antibiotics, several antidepressants block transporters. And so in context, think of how many people are on antidepressants. How many people then will also get an antibiotic, because they've got some infection. And then, whatever level of thiamine they had is certainly not a high level because they're not supplementing thiamine, is now blocked entry. Just the very basic thiamine entry into the bloodstream is blocked. But there's multiple transporters at various junctures that come into play, and any number of them can be affected by pharmaceutical or an environmental exposure or any number of things. And so that places a lot of people at risk of significant thiamine-related illnesses.
Yeah, that's a whole other area to study. I think about chemical exposure. There's a lot of pesticides and whatnot that make their way into our digestive system. And I've not looked at this, and maybe you have, but I'm wondering about mechanisms of certain mycotoxins, which
I haven't looked into mycotoxins either, frankly, but I would suspect that it's the same thing. So one of the things that's interesting about the microbiome relative to thiamine, so most of the bacteria and everything can synthesize their own. I don't know if fungi can, probably can. But what I want to say is that what happens, at least with bacterial balance in the gut, those bacteria that are more adept at salvage pathways for thiamine tend to be the ones that cause all the problems. And so when thiamine is low, that's when you get those pathogenic infections, because those bacterial species can synthesize from byproducts that some of the other ones cannot. And so thiamine is integral to a healthy ecosystem within the gut. And I suspect that is the same with fungi and the molds that we inhale and ingest and everything else, but I don't know.
So let me make sure I'm clear on that. So what are you saying is that if thiamine is low, you've got these opportunistic organisms that, what are they? Would they upregulate their own
They
metabolism?
upregulate because they can make their own thiamine from byproducts that the others produce, and
Oh, well, gotcha. Okay.
and whereas the others cannot as successfully make their own thiamine, because every organism needs thiamine, period.
Right.
So
Is there a class of, let's just say, healthy bacteria, whether it's bifidobacterium or lactobacillus species? Do these tend to? I know there's different,
Okay, so microbiome is not my area, but
Okay.
I have like 3 or 4 papers that I cited in the Hiding Plain Sight paper that tested which bacteria can survive in thiamine versus no thiamine, and that would be a place to start.
Right. Interesting. So, but the take home is that in a low thiamine state, it sounds like you're at risk for, there's the potential of
Yes.
an overgrowth pattern of more opportunistic, potentially pathogenic bacteria. Which is interesting if you think about things like glyphosate, right? So glyphosate, we know, tends to act as an antibiotic to the good bacteria, and then you get a proliferation of salmonella and Clostridia species. So it's fascinating to me, Chandler, that there's this common theme that tends to run across this type of scenario. And so, for me, when I think about this from a functional, integrative health standpoint, looking at the biochemistry, so much of it starts in the gut, right? So it's about of course, diet, certainly the intake, but traditional nutrition, even conventional medicine, takes a big leap and thinks, well, you just need a little bit and then you're fine. But they miss the impact of the gut, the ability to get it into the body, the ability to transport it, and then the ability to actually metabolize it,
activate it. So
right.
Yeah, so one of the coolest discoveries that's happened in the last few years is connecting thiamine to Huntington's disease and a deficiency in thiamine pyrophosphate, which is what activates thiamine. It's the enzyme that takes the free thiamine and makes it thiamine pyrophosphate, the thiamine that you can use. They have a problem with that connection there. So while in the brain, while it appears that they're sufficient in thiamine and everything's going, that enzyme, they can take all the thiamine they want, they cannot activate it into the bioactive form of thiamine that the brain and the body can use. And that's been remarkable discovery, but it shows the complexity of nutrient use, because I'm assuming that there are other examples of that with other nutrients. I don't study other nutrients, so I'm not aware of those, but it's not simply a matter of intake. You can't just say, well, everybody, the NIH says the average intake of thiamine is 6 milligrams a day. So nobody should have thiamine deficiency. And yet tests show anywhere from 10 to 90%, depending on the population, with diabetics having the hardest time with thiamine deficiency because of poor kidney function, because now we know the oxythiamine, but also because they excrete it. They excrete it more readily than other patients. And so you have that. Pregnant women, I'm doing a series of papers for pregnant women, and you think the research in the regular population is poor. In pregnancy, it's particularly poor. The RDA was established for pregnancy, not necessarily based on any evidence that suggested women needed X amount of thiamine to be healthy. It was based upon, well, women are smaller than men and less active, therefore they need less thiamine. So that's the base for women. And then, well, when they're pregnant, they gain a little weight, they're still less active, so we'll add a little bit to compensate. And that was kind of the thinking of that. And most of the studies on setting the thiamine RDA are males, healthy young males. And so these assumptions are rife throughout the research. But even having said that, of the research that came out at the same time as the RDAs were set, which was in the 30s and 40s, researchers were saying, you know, while a milligram or 1.1 milligram is sufficient to stave off the frank symptoms of beriberi, which is, you know, thiamine deficiency, you know, 2.5, 3 milligrams, people can be a little healthier. You know, it's not, and this is just in adults. And but for whatever reason, not just our government, but governments all over said, you know, we're going to single milligrams all you need. And then we'll add a little bit more for pregnancy. And then every year that they review these things, they'll say, well, no new research has come out and we're just going to keep it the same. It's a sad state of affairs. But they ignored research at the time that contradicted their and everything since, frankly.
Well, there's certainly not a lot of money to be made for just promoting, you know,
certainly
B1 intervention or thiamine intervention
or any nutrient intervention.
Yeah, exactly.
Frankly. And so if you stave off the frank symptoms of deficiency, that's it.
Right.
That's all. But you got all the other symptoms leading up to it.
So before we move on, I want to go back real quickly to, and I know this is not just unique to Gardasil, but we go back to that young girl who developed issues, you know, secondary to that. And we talked about.
It was just not her. She was the one that connected me to Dr. Lonsdale. I had we do case reports. Yeah. We had dozens of case reports on the website at this point.
So there's all of these mechanisms, right? There's the intake, there's the absorption, there's the utilization, there's the biochemical sequencing, et cetera. But what I'm understanding, perhaps what you're just mentioning here, is that by and large, for many people, particularly, let's say, vaccine injured, likely the problem in thiamine is the fact that the vaccine and other factors have overly stressed and put increased demand on mitochondrial function to even just maintain normal cellular function.
Yes.
And so the system is highly stressed. And if somebody was even somewhat deficient, now that just becomes fully expressed. And those highly metabolic areas in the body, neurologically, brain nervous system, cardiovascular system, are usually the first places to take a hit, you know, from a clinical standpoint. Does that
yes.
make sense? Okay. So that potential then exists, as you mentioned. It's not just Gardasil. It could be many other vaccines. It could just be other compounds, other pharmaceuticals, other toxicants, for example. The more these things pile on in various ways, they affect and overstress the system. We're putting increased demand on these nutrients. And one of the things, Chandler, that I've seen a lot I do a lot of organic acid testing, and so through my practice. And one of the things I see on that test, there's two areas, by the way, I want to bring up. One is riboflavin. So I see a lot of people with vitamin B2 deficiencies. And when you look at the biochemistry, it's incredibly complex too. And so I'm thinking, okay, there's got to be just, again, this increased demand, because it does play an intimate role in cellular metabolism, particularly at complex 2. One of the other things I see on that test is an elevation of succinic acid. And one of the things that I read in your paper was, we know that thiamine plays a rate-limiting step in pyruvate dehydrogenase complex, in alpha-keto dehydrogenase, the branched-chain keto acids. But you also mentioned that thiamine sort of, and maybe there's other enzymes too, although it's not specifically acting as a cofactor in succinic dehydrogenase reactivity, a deficiency state, I don't know if it puts pressure on the enzyme, if it somehow alters the chemical nature of how succinic dehydrogenase reacts. Is that something that you remember or can speak to? Because I think that's incredibly important too, to understand it's not just a direct effect, but sometimes we get indirect effects.
Let's start with the first question, riboflavin. Sure. Okay, so riboflavin is typically depleted for a couple reasons and important for a couple reasons. Riboflavin is depleted with arsenic and mercury exposure, which the food chain, everything is loaded with that, medications, etc. But riboflavin, why, in addition to its role in the electron transport chain and in the TCA, it is a cofactor, or is a vitamin cofactor in the PDH with thymine. So it's three subunits of that enzyme. One that requires thymine, one's riboflavin, there's some other things, and the other is alpha lipoic acid. And tangentially, I'm going to mention this with regard to alpha lipoic acid. One of the treatments, of choice these days for people with diabetes is to give high doses of alpha lipoic acid. And if you do that and the person's already found with thymine, you're going to shut the whole thing down. So totally against high doses of alpha lipoic acid. Just saying that. So that's question one. Question two, with regard to allosterically regulating other enzymes that it may not be directly involved with, I think that just makes sense, frankly, because again, its role in energy production, if you start blocking things that involve the capacity to produce energy, most of the reactions are going to downregulate. And the enzyme functions, they may upregulate initially to compensate, but over time are going to downregulate relative to the diminished ATP capacity. And so it doesn't necessarily have to have any direct role with thymine itself. It's just a response that the body's going to make to the capacity that it has available to do what it's got to make decisions. And so everything's going to get downregulated to some extent in that whole chain.
Right. That makes perfect sense. Thank you for that. So let's shift real quickly and just talk about for the listener. So, thiamine obviously gets tagged to beriberi, for example. But in your work, in your research, and in your writings, I see a lot of need in many of the special needs kids, the autistic kids. We know that other patients with metabolic problems certainly as well. And we could extrapolate this out even to some of the neurological disorders, whether that's Parkinson's or Alzheimer's or whatever. But what should practitioners and maybe even just the general public sort of be aware of, or at least have a suspicion of, that thiamine deficiency may be a contributing factor, play a role, perhaps, in their particular condition, or in practitioners' cases, particular patients?
I would think it plays a role in just about everything, and perhaps I'm biased. But if you have a conglomeration of conditions that make absolutely no sense, then thymaine's involved in it, because
Right.
mitochondria are involved in it. And whenever mitochondria are involved in something, mitochondria influence everything, frankly. But mitochondrial disease, even genetically determined mitochondrial disease, never present in a consistent phenotypic pattern. You can have siblings and an entire family with the same genetic issues, and all of them will have entirely different scope of symptoms. And this is because it interacts with so many other factors, the environment and stressors and how your body adapts to it, your nutrient levels, all of that. And so it's going to express itself. Mitochondrial disease and mitochondrial deficiencies, if you will, are going to express in a highly varied context. Certainly with neurological issues, certainly with gut issues, certainly with behavioral issues, which are essentially neurological issues, regulation of sugars, regulation of, everything is going to be affected because mitochondrial capacity is insufficient to meet the demands of daily consistent living, and so any stressor that person or that body is presented with, compensations have to occur, and those compensations are what are causing the symptoms that you see. And so for children, I think that it would be in our best interest to begin looking at nutrient capacity, mitochondrial capacity, thiamine capacity much earlier, particularly with the number of stressors that they are put under from a very early age because of the way we manage our vaccines, but also from birth. If we look at mom and her health during and prior to pregnancy, and even dad, because he contributes to this mess as well, if the parents have issues going in, the child, even if the pregnancy is healthy and normal, so to speak, the child is going to have issues coming out with mitochondrial function. And so we need to start looking at this through a broader lens, frankly, and not looking for, oh, if it's this, it must be that. It's not an X to Y relationship. It's the mitochondria are taxed and suffering. So we need to support the mitochondria so then they can start unwinding everything that they've done to keep you alive. They do the things they do, and then I think we can be healthy. But just simply an X to Y, or, if he has these symptoms, it must be thiamine. If he has that symptoms, he must be thiamine. I think it's much broader than that.
Yeah, I love that. And that's the way I tend to operate, is to look at things globally, to try to make those connections, but to understand it's not just one particular thing, it's how these things interrelate. And you're right, when you get down to it, so much at the fundamental level is what's happening within the mitochondria. I like the example you mentioned about, the different types of, genetic mitochondrial problems that run in a family, and you can have 4 different people that have entirely different, presentations.
Yeah.
And also the other thing too, and I picked it up from your paper, we think about, right, the intake of thiamine, right, begins in the gut. Well, you can start to manifest with gut problems primarily because the mitochondria in the digestive tract aren't getting,
sufficient
fed
thiamine.
sufficient
And
thiamine.
I think
So
that's
that
a
could be
key
a first
point for all of the GI disorders we experience these days. It's just GI disorders are huge. And people get to the point of completely severe intolerance because, bottom line, they were deficient in nutrients to begin with, continued to be deficient, and now the mitochondria in the gut do not have the energy to actually digest food, and so it just becomes this never-ending cycle that gets worse and worse and worse. And I see people with food intolerances are down to like 4 foods, and that even before they got down to 4 foods, they were deficient, but now they're deficient in everything,
Right.
and you cannot heal. You can't throw pharmaceuticals at that and expect them to heal. And yet that's what they continuously do. They throw, this medication, that medication, and then all the while the person is starving to death, literally. And so, yeah, heal the gut and you will probably heal quite a bit of everything else.
Well, take it one step further, and what you mentioned about thiamine deficiency in the gut, you get an expression of pathogenic, problematic bacteria. Now you get a depletion of diversity in the healthy microbiome. That sets the stage for other factors like fungus. And so, you get fungal
and you
expression.
also get histamine and mast cell degranulation, because whenever the cells are deficient in thiamine, you get degranulation of mast cells. And so there's your Macus reaction. There's that everybody has. But the problem is how do you replete these people with thiamine? One, when you can't do IV 98% of the time because no one will give IV thiamine anymore, and 2, they can't tolerate. They literally will vomit or, have a reaction to the slightest bit of food. And we saw that, citing the paper again. There were a series of papers that I discussed in the Hiding in Plain Sight paper that were based on studies done in the 30s and 40s in a female psychiatric patient population, where they controlled food intake, controlled nutrient intake, and removed thiamine. They put these women on very, very low to non-existent thiamine and tracked their symptoms across periods of time. Some of these women were getting 0.4 milligrams of thiamine per day for weeks, if not months. And what they found was a couple interesting things. The more active people got sicker faster. The leaner, more active, presumably healthier, got sicker faster. So if you were larger, it took longer. You had more stores, presumably. But most of it went to the gut. They would begin not being able to eat anything. And it got so bad in a couple of the patients that they would vomit even in response to parenteral feeding. They couldn't, the food intolerance was just intense. And so what that tells us is that the gut plays a huge role in it. And as we see systems like gastroparesis and SIBO, and you see IBS, depending on which direction you go, you start seeing those things. You got to start thinking, not only is the microbiome dysfunctional or screwed up, but ask the question, why? Why is the microbiome the way it is? Did they all of a sudden become exposed to all kinds of different bacteria that landed in their gut? Well, that's possible, but more likely is the bacteria don't have what they need to do the things they need to do to metabolize the food and send it out. And so that's why you're getting those reactions. But that series of studies, if you go to this paper and then look up that series of studies, they're remarkable in their discussion of how these women presented the symptoms, how the trajectory of symptoms. None of it looked like what we would think traditional beriberi is. And none of it happened in a manner, a speed in which we would have anticipated or we're taught to think it happens. Some of these women lived all of them lived, but were surviving for 6 months on these exceptionally low levels of thiamine. They were seriously ill, but they were surviving. And so in our textbook understanding of thiamine is that it's acute illness and that you need to be hospitalized immediately and you're going to die or brain damage. But that's not the case. And it's particularly not the case as we've gotten away from, starvation, because our thiamine levels are going to wax and wane, across time. You're going to go for periods of time where you're deficient and periods of time where you're not. And the body adapts.
Right.
And keeps you alive. But I think the gut is very important. And I think when you see excessive vomiting or you see excessive food intolerance, you've got to start thinking thiamine and other nutrient deficiencies, not as a consequence, but as a trigger.
So, a couple of thoughts on that. And one thing is, here's your paper, and in the description of this video, once we get it posted, I'll make sure to create a link, because this is actually a great paper. I really encourage people to go find this, download it, read it. There's a ton of references towards the back. Just a great, and this is actually a great segue into your book with Dr. Lonsdale. One thing I think about, just think about what we've been talking about, mitochondria. So just think about the gut, right? So every cell has mitochondria. Red blood cells don't, but every cell, other cells have mitochondria. So in order for an epithelial cell to absorb nutrients, it needs energy. In order for a cell to help secrete secretory IgA, it needs energy, meaning mitochondrial energy. For a cell to produce mucin layers in the gut, it needs energy. So every cell in our body that has a role, our immune cells, our endocrine cells, our heart cells, whatever, it all is dependent on ongoing mitochondrial activity, which means it is dependent on ongoing nutrients like thiamine. So when you start breaking things down in that regards, you can start to see what you've been discussing here about how dysfunction can manifest. I find it fascinating what you mentioned about mast cells, in that I take that as mast cells become more sensitive to the release of histamine in a thiamine deficient situation. So there must be something within the mast cell, mechanism that in regulating histamine reaction is mitochondrial dependent to some degree. So at least that's the way I think of it.
Perhaps. Yeah, I think it is. I've always been interested in finding the cause of things,
right.
The pathway and the cause. And I think mitochondrial function or dysfunction is fundamental to just about everything else. I mean, I don't know if you can go to a lower level, a more fundamental level than mitochondrial function. That's as far as I've gone. But we never talk about that in modern medicine, interventional medicine, and even in functional medicine, we don't look at mitochondria. I see, as I said, on our website, Hormones Matter, we have hundreds of case stories at this point. Patients write their experience. And I see so many of them who have been through, dozens of traditional doctors and dozens of functional doctors, and none of them have really addressed the core components of their illness. The flaw with a lot of the functional medicine is to take the model of conventional medicine and except use nutrients or other herbal supplements and such. And I don't see them doing it, and this is not everyone, but I don't see them addressing the weaknesses that patient has. So in one of our Facebook groups, Understanding Mitochondrial Nutrients, we will regularly see people post their oat tests. And the patient will say, my doctor prescribed this and XYZ is either high or low, but she doesn't tell me anything about it. Nobody's told me what it means or what to do about it. What do I do about it? So I see this testing, and then they will throw a lot of nutrients or herbal supplements that are not matched to the oat test or to that patient. It's just like a standard protocol. And so I think we got to get back to looking at the patient as an individual, looking at what they're presenting with and how that is affected or being affected by the mitochondria, how the mitochondria are affecting those symptoms, and kind of back down that pathway to figure out what they need. The variety of testing you do can point you in those directions, but you got to do something with it, and you got to correlate that with the patient's experience as well to kind of put it all together. And I just don't see that a lot at all.
One of the things that I emphasize in my training courses is, every person is unique and every person's an individual that needs to be treated that way. Every test you do needs to be applied clinically to the clinical presentation of the patient. And I actually stole this from my partner. Standard protocols are a place to start, but they're only as good until the next person they don't work on.
Exactly.
And then you have to figure out where to go next.
And that's the other thing. People will ask you, well, what's the protocol? And I was like, there is no protocol. What do you need?
That's exactly right.
Nobody wants to look at that. I mean, certainly you can put a protocol together and it's useful to have a reference point. But as far as I'm concerned, there's too much variability between people to do that. And so, that's my opinion. But I'm not a practitioner, so
with the remaining time left, I want to make a quick comment. I want to ask you two additional things, and then I want to show you a website. So as you were talking, one of the things to get across, and I want practitioners who are listening to this to understand this, and I want the general public, if they're listening to, is that mitochondrial dysfunction, you have genetic things that might get expressed across sort of the general platform of the body, but it mostly becomes manifest in those high metabolic areas of the body. So again, brain nervous system, cardiovascular system, immune system, things that these organ systems
but also
that demand.
Musculature and
absolutely
kidney and things like that. So,
absolutely. So, but I guess the point is that you can have mitochondrial issues that may be expressed in a specific localized area in the body. To take the digestive tract, for example.
Yeah.
It doesn't always, at least obviously, sometimes manifest as this something that is easily recognizable for some people all over the body. So it could be somewhat compartmentalized. It could be highly concentrated in the kidneys or the liver or the digestive tract or whatever, which sort of gets to the point that the body is pretty adept too at localizing, compartmentalizing things for a period of time. Some of it we control, some of it we can't. As you mentioned before, fundamentally get down and just understand, what do mitochondria do and what can we do to support them. At some point, right, the inherent wisdom of the body and all of that is going to take over and do what is necessarily going to need to do to kind of help. So two additional questions I want to ask. One, laboratory testing. And this always gets to be a big one, because I know that there's not always going to be that one test that provides the sole information. But can you give a little bit of an overview on sort of the common available tests that might be now that give perhaps some insight, into a potential?
So yeah, there's serum and blood testing, and that's plasma testing, excuse me. That's the most common. Those are highly inaccurate and typically have a high false negative rate, because they're reactive to what you ate the night before, and your current status. However, having said that, if you test low on one of those, you have been low for a very long time and you need to get treatment. And also, having said that, most doctors ignore that. And I hate to say that, but I have cases where the patient was tested low, and they were tested low years ago. No one ever mentioned to them until they get to another doctor, like the 12th doctor in a row, reads the test and say, oh, you're low in thiamine, and have been for the last 5 years. So again, that goes to our classical description of, you're going to die, of severe illness immediately after becoming thiamine deficient. You can live for years because it sort of will wax and wane, but you can be low for years, but you're going to be severely ill. So there's that. There's whole blood testing, which is a little bit better. And depending on who does it and what method they use, you can get a little bit more accurate test. There's also a functional enzyme test called a Transketolase test, or erythrocyte transketolase activation test, and only a few labs do those, although I'm working with a foundation and with someone else, individual who has developed some more transketolase tests, and we're hoping that at some point in the next year they can bring out more transketolase testing, because
good.
that's one of the more accurate ways to test. Interestingly, I also just read a paper and wrote a quick little blurb about it on the website, about a paper was in 1969. But what they did is they did a pyruvic acid challenge test to identify thiamine deficiency. This was in preeclamptic women. So pyruvic, or pyruvic acid, has a narrow range of values in normal people. And even when you challenge it with dextrose, in folks who have sufficient thiamine and everything else is going well, there's a range, an acceptable range of increase. In those who are low in thiamine, depending on the stage of deficiency, the early stages, they look okay when you take it fasted and there's no dextrose. But when you add dextrose, it spikes, kind of like your gestational diabetes test. It spikes. In women that had severe preeclampsia, it was elevated both fasting and then spiked even further post dextrose challenge. And I thought it was a unique way to address this. And I was talking to a lab friend of mine, and he's going to look into the possibility of seeing if they could design a clinical test like that, because that would account for all sorts of folks who appear to have normal thiamine concentrations, but with high carbohydrates, which is the diet, you will be able to see with the dextrose challenge that they don't have sufficient thiamine relative to their needs. And so I think that was a unique way to get tested. So those are the main tests. They all pretty much are abysmal, because either you can't get to them or they're inaccurate. And in fact, in acute thiamine deficiency in the hospital, they tell them to treat regardless, because it takes too long to get the test and the test may not be accurate. But so in most cases, people are left to treat clinically, because thiamine is safe. and you will excrete what you don't use. So you can take relatively high doses without a problem. And so oftentimes people will treat clinically. If you think you have thiamine deficiency, they will take thiamine.
And so that's kind of I think that's really the take home point. The tests are spotty. So,
yeah,
if you're low on a test, you're deficient. If you're normal
yeah.
on a test, you still could be deficient.
You still could be deficient. Now, I think
Yeah,
you talk about oats test, and so that's a test where you get to see patterns,
right.
and there are certain patterns that can point to a thiamine deficiency, a prospective thiamine deficiency, but not everyone has access to an oats test.
Yeah. And
And
they're
so,
patterns. There's not a marker that is, one
yes,
marker that says, oh, thiamine, you're low, here's the marker. There's not that. You have to actually
yeah,
look for patterns.
yeah.
The other question before we wrap up, and this obviously could be another discussion, but different
Mhm.
forms of thiamine from a supplement standpoint. And we're not getting into treatment, we're not giving treatment recommendations, but just sort of, overall, I mean, I know that there's different forms. Some may be more absorbed or more easily absorbed, from a general standpoint. Your thoughts on that?
So yeah, so there are different forms. So like thiamine hydrochloride is the standard that you get in most over-the-counters. Thiamine mononitrate is in the cheaper over-the-counters, and I probably wouldn't recommend that, but hydrochloride is fine. It's not as well absorbed as we'd like it, and so you have to take high doses of that. But because it's not absorbed particularly well, it actually works okay for some folks who are highly reactive. And so they can dose that one more easily than they can, say, a thiamine that has TTFD in it or benfotiamine. The TTFD brands are allithiamine, lipothiamine, and Thiamex.
And those are
Thiamine,
sulfur forms, correct?
yes, those are sulfur forms, and
Okay.
they're very highly absorbed. They don't require transporters. So if you happen to know that you have issues with transporters, you can use those, as benfotiamine does not require a transporter either. But they can be too potent for some people and or people who have problems with sulfur. And most people with problems with sulfur are actually thiamine deficiency. So it's the thiamine deficiency. So it's that cycle. But getting to the point where you can handle that is another question. So sometimes they have to start with microdosing of thiamine hydrochloride and then move on to. Now, having said that, in some cases, benfotiamine seems to work really well with people with GI disturbances. I don't know why it works better with GI than others, but it does. The thiamine hydrochloride seems to work quite well with some people who have chronic fatigue. And the TTFD works well with just a lot of different people, but it reacts with some people. So you have to, you have to experiment, frankly, and figure out which one works best for you.
Yeah. And that makes perfect sense. It's like anything, it comes down to the individual again, how do they react? How do they respond? And there's some general rules, as you mentioned, but by and large, it's doesn't always fit, right? There you go back to the protocol. The standard protocol is only as good until the next person doesn't work on, and you got to figure out for that individual. Hey, by the way, I just thought of something, and when you're talking about the thiamine hydrochloride being relatively poorly absorbed unless you're in really high amounts,
or IV,
or IV, any thought on if it's being more retained in the digestive tract? Could that then lend towards some actual benefit on the microbiome for the good bacteria because it's not passing through so quickly? So there's more getting down, particularly down in the large intestine.
Interesting. Yeah, I know. I hadn't thought about it. That's a possibility.
So yeah. Anyway, that's another interesting area potentially.
Another nuance that one has to figure out. Yes.
So Chandler, where can people access your work? I showed your, let me bring up my screen. So I highly recommend people read your book. So this is available, I'm assuming, through most distributors. This is just a screen capture from Amazon.
Yeah, Amazon is where it's easiest available, or the publisher elsewhere.
Okay. And then Hiding in Plain Sight, this was actually, I believe it's free as a download online.
Yep, it's free download, open access. We'll only ever publish in open access.
Okay.
I don't agree with paywalls.
And then you also have another website. This is your Hormones Matter matter.com website. Tell me real quickly about this site. What's this about?
Well, as I mentioned, I started out with women's health and hormones, went to medication adverse events, primarily in women, and then eventually landed in the mitochondria. All the while, the core tenet of this was that we would have case stories, because I think that self-told case stories provide us with information that we don't get in the case studies that are published through the lens of someone else. And so we allow patients to tell their story. Those stories often point me and others in directions of research that I didn't know. I find out medication reactions and have done studies on that based on a particular case study that came up and said, hey, I reacted to XYZ. What do you know about it? And then I go in and I investigate and I figure out that these mechanisms were involved in his or her reaction. So my goal is to build the knowledge base of these kind of more complex issues relative to general health that don't fit in any diagnostic category that is currently published. So that's it. So I publish a couple new articles every week. It's either from researchers, physicians, and or patients who are either experts in an area or experts in their own health, and they just present their case study. So it's a wide variety of authors. And at this point, I think we have about 1,600 articles on the website. So there's some depth to it.
Yeah.
So that I've been doing for the last over a decade, just because I enjoy looking into things and learning. And that is my vehicle for learning.
Well, you're a kindred spirit. I'm the same way. I have an appetite for learning, and that was one of the things I appreciate about your work. I really have. I've learned so much from your website, as I mentioned, your books, your articles. So it's very much appreciated. And any final thoughts? Any words of encouragement out there that you want to?
Well, I think everybody needs more thiamine than we're getting. So just keep that in mind. And I would like people to begin trusting themselves and becoming, I don't want to say their own doctors, but their own advocates for health. If something's wrong, it's wrong. You know it. You have the power also to figure it out and change it. So, take the time to learn these things, what you can. Some of it's complex, but at least on the website, I work very hard to make everything digestible for the layperson. Sometimes I don't succeed, but my goal is to make some of these pathways and these mechanisms more readily understood. And so you can do it. So if you are ill or your child is ill or your spouse is ill or something like that, do the research. It's all out there. You can figure this out yourself. It may take some time, but I think you have a pretty good chance of figuring out how to heal yourself.
Yeah, very true. Well, Chandler, thanks so much for joining us.
Oh, you're welcome. Thanks for having me.
You've been listening to the Functional Medicine Doc Talk podcast with Dr. Kurt Woeller. For more information about this podcast, go to functionalmedicinedoctalk.com.